So, welcome to our deep dive into the fascinating world of bacterial RNA polymerase. In this video, we are uncovering the intact details of one of the most essential enzyme is biological responsible for transcribing DNA into RNA. And today we are going to explore the structure, function and significance of bacterial RNA polymerase. E. coli RNA polymerase has a unique shape resembling a crab claw with a projection flanking a cylindrical canal. This canal is sized to directly bind 16 base pair of DNA. When the polymerase binds to DNA, it covers a region called the promoter, which span about 60 base pairs. Within the region, 16 base pairs are situated within the active site where transcription occurs. The activity of E. coli RNA polymerase relies on magnesium ion, which with a transcription rate of approximately 40 nucleotides per second at 37 degrees Celsius. The overall structure RNA polymerase enzyme across different organisms resemble a crab claw. The two largest subunits such as beta dash and beta in bacteria or RNA polymerase beta 1 and RNA polymerase beta 2 in eukaryotes. From the pinces of the claw with the active site located at the base of these subunits. The active site with the active center clipped of the polymerase bind to the magnesium ions, which are crucial for its function. The requirement of magnesium is common among various types of RNA polymerase. RNA polymerase features entry and exit canals that facilitate passage of DNA, RNA and ribonucleotide into and out of the enzyme's active center clipped. These canals play a significant role in the mechanism of transcription, which will be explored further. So now we need to understand different structure of E. coli RNA polymerase. The Escherichia coli RNA polymerase is a multi-subunit enzyme that's made up of 5 subunits, 2 alpha subunit, 1 beta subunit and 1 beta dash subunit and 1 omega subunit. The alpha subunit are identical while the beta and beta dash subunit are large. The omega subunit has a molecular weight of 10 kilodalton and it's required for cell viability for enzyme activity in vitro condition, while association of sigma factor makes the hollow enzyme. Now here we discuss about each and every subunit of bacterial RNA polymerase. So first is beta dash subunit. Beta dash subunit, the largest subunit of RNA polymerase, is produced by the ROPC gene. Within the beta subunit, there is a crucial part called the active center, which is accountable for RNA synthesis. Additionally, it contains elements that facilitate interaction with DNA and newly formation of RNA molecules. Hairpin, a type of polyanionic, can be observed to attach to the beta dash subunit. Hairpin's presence inhibits the process of transcription in controlled laboratory conditions. Furthermore, it completes with DNA for bonding with the polymerase. These findings imply the beta dash subunit likely play a role in attaching to the template DNA during transcription. In its function, the beta subunit holds onto 2 magnesium ions, which are believed to aid in the catalytic activity of polymerase. Interestingly, in cyanobacteria and chloroplast, the beta dash subunit undergoes a division into 2 separate subunits. And now we will discuss about beta subunit. The beta subunit encoded by the ROPB gene is the second large component of RNA polymerase. Within the beta subunit lies the active center crucial for RNA synthesis. It also holds elements necessary for interaction with DNA and newly forming RNA molecules regardless of their specific sequence. Structurally, the beta subunit comprises 2 distinct domains. Rifampicin, an essential antibiotic, target RNA polymerase by binding specifically to the beta subunit. This binding interacts with the initiation stage of RNA synthesis but doesn't affect elongation. Unlike many antibiotics, rifampicin does not hinder eukaryotic polymerase. Consequently, it finds extensive use in medical treatment, particularly against gram-positive bacteria infection and tuberculosis. Another group of antibiotics known as Streptolydigins functions by impeding the elongation phase of transcription. The differential impact of these antibiotics on a single subunit, the beta subunit, suggests that it likely harbor distinct domains responsible for both initiation and elongation phase of transcription. And then let's we detailed understand about alpha subunit. So alpha subunit, the RNA polymerase has a alpha subunit which exists in two forms, alpha 1 and alpha 2. Each alpha subunit has two key parts, the N-terminal domains and the C-terminal domains. The N-terminal domains play a role in assembling the core RNA polymerase. Bacteriophage T4 alters the alpha subunit by adding adenosine diphosphate ribose to an arginine. This modification changes the shape of the alpha subunit, reducing its ability to bind effectively. This modification suggests that the alpha subunit is crucial for binding the promoter. Specifically, the C-terminal domains of the alpha subunit interacts with promoter DNA. It engaging in non-specific interactions with most promoter and can also engaging in a specific interaction with promoters containing upstream element. Additionally, the C-terminal domains of the alpha subunit is responsible for interacting with regulatory factor, indicating its role in coordinating gene expression. And our fifth subunit is omega subunit, which is very important role in RNA polymerase during transcription. So now we need to understand omega subunit. The omega subunit, being the smallest subunit in RNA polymerase, play a crucial role in RNA polymerase. Its main job is to help assemble RNA polymerase effectively. Additionally, it contributes to stabilize the assembled RNA polymerase complex. And the last and most important subunit is a sigma factor. E. coli RNA polymerase holoenzyme consists of several parts, including a key player called the transcription initiation factor, also called sigma factor, with the core enzyme. Sigma factor's job is to make sure the RNA polymerase knows where to start transcription. The complete holoenzyme is made up of 6 subunits, alpha subunit, beta subunit. In this work together to ensure accurate transcription. While there's only one core enzyme, different sigma factor can team up with it to transcribe different genes, allowing to a specific targeting. Sigma factors is like a navigator for RNA polymerase. It helps the polymerase find the right spot on the DNA to start transcription, reducing its attraction to random DNA, bites and enhance its affinity. For specific promoter regions, this ensures transcription kicks off in the right place. Once transcription begins, sigma factor's job is mostly done. It's like a guide who steps back after leading you to the right path. Initiation, sigma factor disengage for holoenzyme, leaving the core to carry on with the essence of transition. The core enzyme without sigma factor is still capable of elongation the transcription. So after initiation, the core enzyme is all that's need to continue to the transcription phase, produce DNA and adding nucleotides to the growing RNA chain. Now we summarize all these factors. Subunit functions. First is a beta subunit encoded by ROPC gene. It contains the active center for RNA synthesis and interact with DNA and RNA. Binds to magnesium ions essential for catalytic. Hairpins can inhibit transcription by binding to this subunit. And second subunit is beta subunit encoded by the ROP beta gene. It contains elements for interacting with DNA and RNA. Antibiotics like Rifampicin and Streptolydigins target this subunit to inhibit transcription at different stages. And third is a alpha subunit, consists of N-terminal and C-terminal domains. The N-terminal domains involved in assembling the core enzyme and the C-terminal domain interacting with promoter DNA. Regulatory factor modification by bacteriophage T4 can affect binding effectively. And the fourth subunit and the last subunit is omega subunit, a smallest subunit crucial for the assembling and stability of the RNA polymerase complex. and one of the most important factor is a sigma factor associates with the core enzyme to form the hollow enzyme, directing RNA polymerase to a specific promoter region to initiate transcription. Detach after initiation, allowing the core enzyme to elongate the RNA transcription. And the most important part of our video series is a related fact important term is bacterial and yeast RNA polymerase share similar shape and organization. Antibiotics like actinomycin can block transcription for a double-strand DNA template by both E. coli and yeast. RNA polymerase because of their shared similarities. This structural is more pronounced than their sequence similarity, especially in the internal region near the active site. Archaea have a single type of RNA polymerase responsible for synthesizing all RNA. Archaeal RNA polymerase is particularly closely related to the eukaryotic nuclear RNA polymerase. Thanks for watching. See you in the next video.